afternoon, everyone, and welcome to today's Public Health Ontario Microbiology Rounds presentation on missing gut microbes and where to find them. My name is Melissa Richard Greenblatt. I am a clinical microbiologist at Public Health Ontario, and I have the pleasure of moderating today's session. So before we get started, I'll mention a few housekeeping items. To enhance the presentation experience, participant audio and video has been disabled. The chat pod has been deactivated to limit any distractions during the presentation. Please use the Q&A pod if you have any questions during the session. A discussion and question period will follow the presentation. And if at any point during the session you experience any technical issues, please email capacitybuilding@ohpp.ca. I would like to state that as the moderator of this session, I do not have any conflicts of interest to disclose. So it's now my pleasure to introduce the speakers for today's presentation. Emma Allen-Vercoe. So Dr. Emma Allen-Vercoe is a professor and Canada Research Chair at the University of Guelph, where among many other things, she studies gut microbes that are volatile or associated negatively with industrialized societies of humans, and this is called vanish microbes. So now I'm going to pass it off to Emma. Thank you.
All right, well, thank you very much, and thank you for having me. It's lovely to be here, and I'm very excited to talk about some of the really exciting stuff that's going on in my lab. And thank you for the introduction. And yes, so today I'm going to be talking about missing gut microbes, and these are microbes that we are particularly interested in, that we're finding in some really unusual places. So let's move on from there. All right, so before I start, I want to just point out that I am a co-founder and the chief scientific officer of Microbiota. It's a company that we created about 10 years ago to create microbial ecosystem therapeutics to treat a range of indications with gut microbial imbalance as the root cause. And I will mention the work of this company at a very, very high level in my talk today. All right, so before we really get into the meat of things, a few introductory slides, and I'm guessing that many people on this webinar will appreciate this, but this is a very important point, that we are not just human. We are in fact complex superorganisms of human and microbial cells. We exist in a delicate host microbe equilibrium. That word delicate is a very important one, because we're realizing more and more that that we've been doing damage to that ecosystem, and I'll talk about that shortly. Now, most of the microbes that live on and in our bodies actually reside within our gut, and in particular, they reside most densely in our colons. Each gram of feces contains around 10 to the 11 bacterial cells. If you take a minute to think about how many zeros that is, that's an awful lot of cells. And that's those cells, and those are bacterial cells. They represent around about 200 to maybe 500 different species. 200 usually in the industrial world, and I'll get to that in a minute. But as well as that, there are also other types of microbes that live in our gut. I'm not going to be necessarily talking too much about them today, but we shouldn't forget that they also contain, or the gut also contains archaea, sort of ancient type of organism, which are actually look like bacteria, but nothing like bacteria biochemically. And also viruses and yeasts and fungi and some protists as well. So all of those things we're considering. Those are very important parts of the gut microbiome. But today I'm just going to focus on the bacteria, and in fact that's where most of the work to date has been done, and where we probably know the most. So how human are we? A few years ago, you might have seen a reference to the ratio of being 10% human. So in other words, you're only 10% human, and most of you is actually microbial. Well, a few years after that, Sender and Fuchs and Milo actually published a paper after some very careful calculations, where they created a reference human of around 70 kilos, 20 to 30 years old, 1.7 meters tall, and did some very fancy calculations to figure out how many human cells such a person would have. And it turns out that they have around 30 trillion human cells. And if you look at the number of bacteria that live on and in us, That's around about 39 trillion bacterial cells. Difficult to measure because at any one time that might change, for example, after a bowel movement. But for the most part, 39 trillion bacterial cells. So they made a calculation that we are in fact 1 part human to 1.3 parts bacteria. So although bacteria are still in the majority in terms of cell numbers, they're certainly not as high as 90%, which was what we heard before. And this is probably more in line with what many of us were thinking when that first idea came out. And as I mentioned, there's approximately 200 different bacterial species per person. And I'd like to show this image. This is just a scanning electron micrograph of a corn kernel that we took in our lab a number of years ago. We retrieved this from a human poop sample, and I love this image just because it's very pretty for one thing, and I love looking at scanning electron microscope images. But second of all, just to point out that although you might think of bacteria as sort of inanimate objects, little sort of blobs of goo, in fact, they're actually quite different to each other. When you look at this level, you can see that there's lots of different types of cells here. They have different cell wall structures. One of them looks like a peanut in the middle there. I love that one. But they're all also, if you look very closely, rather connected to each other with sort of protrusions that come out of the cells, and that's quite common as well. And I think that's kind of illustrative of the fact that these microbes form a community, and that's a very, very important point to make. They're not just inanimate objects that just float around doing their own thing. They are very much interconnected with each other, and together they actually form a superorganism, which we'll talk about in a second. So another introductory slide, another very important component of microbial ecology is that biodiversity in the gut is extremely important. When you have a high diversity of species, just like you have in a rainforest here as depicted in the image on the right, you have a robust ecosystem. And in that robust ecosystem, you have balance, and the balance is due to the fact that you have functional redundancy. And what functional redundancy means is that essentially you have a large number of collective genes, and those genes can be shared amongst a number of different organisms. And this is true at the microbial level as well as the macro level. When you have that high gene count, it means that you have resistance to damage. Because if you were to take one species out of this ecosystem that you see here in this rainforest, and maybe if I told you to close your eyes first and I took one away, it might take you a beat to figure out which one I'd actually taken away. And functionally, you probably wouldn't notice much difference because there are lots of other organisms that can step up and take the part of the thing that's missing. Now, if you contrast this to an ecosystem with a low diversity of species, that's a much more fragile ecosystem. And the example I have here is an image from Kananaskis National Park, which is just in near Calgary, between Calgary and the Rocky Mountains in Alberta. This is an example of a naturally fragile ecosystem just because of its geography. It lives, it's a very high space in terms of high altitude. It's a lot of UV light from the sun. It's very dry and very cold in the winter and quite hot in the summer. And as a result of that, there are very few, compared to the rainforest, let's say, there are very few organisms that can live there. And as a result of that, there's imbalance in the system from the point of view that if something happens and that system is perturbed, then there's a likelihood of a collapse of one or more of the species that are present. And this happened a number of years ago when climate change allowed the movement of a pine beetle across the Rockies from British Columbia. And they decimated the white pine population in Kananaskis National Park, and that's what you see there. And this is just an example of an ecosystem with a fragile functional disability that is very much more susceptible to damage. And so I'm talking about forests there, but just as at the macro level, microbes and the rules of ecology are the same at the micro level. And so that's just an analogy that I've drawn. Now, everyone has a unique microbiome composition in terms of species. So you and the person sitting across you in the room will probably have a very different microbial species composition. But what we've learned in the last 10 years or so is that it's not really the species that you have in your gut, but it's what they're doing with their metabolic potential that's important. And these two pie charts on the right-hand side is from a paper that actually compared the gut microbial composition from 1,004 twin pairs. And what they showed on the left-hand side, when you look at the species composition, only 8% in the yellow part of the pie there, only 8% of those species are shared or detected in more than 75% of the samples. And so you can see just how different people's gut microbiomes can be from each other. But when you look at the pathways that those species actually enable, that they code for, what you see is a very different picture. And on the right-hand side, you can see that 57% of their pathways are actually detected in more than 75% of the samples. And that's a direct result of that functional redundancy that I just mentioned. So what do our gut microbes do for us, and why am I so focused on the gut microbiome? Well, they regulate the immune system. They do many, many other things as well. That immune system regulation seems to be one of the things that most people are focusing on right now for good reason. It does play into what I'm going to be talking about shortly. They also obviously help to extract the energy from the foods that we eat. They control potential pathogens, and in the past it used to be thought that they just controlled potential pathogens by crowding them out, and now we know that it's a lot more delicate than that, and in fact that they're pumping out all sorts of molecules that naturally prevent pathogens from behaving in virulent ways. A lot of these gut microbes make some essential metabolites, including vitamins and cofactors. They improve intestinal function. They remove toxins and carcinogens and from our diet and the environment in which we live. And so collectively, and I've just taken a very small number of the functions there are many more. Collectively, these functions mean that the microbiome is as important to us as a liver. And really, we should be thinking of it as a virtual organ. And just like a liver, you can't live without your liver, we should be thinking a lot about how we shouldn't really be living without our microbiome either. So how do we acquire these microbes? Well, it's commonly assumed that the fetus is actually in a bubble, the amniotic sac, if you like, in a sterile environment. This is a fairly controversial topic, but I think it's being resolved now, and really that is shown to be true, except during infection, which is an unusual event in this case. And so the fetus doesn't actually really come into contact with microbes until the process of birth. And so imagine that this fetus is moving down the birth canal, it's picking up a lot of microbes as it does so. That process of the fetus moving down the birth canal, as anyone who's given birth knows, can take some hours. And during that time, that fetus is swallowing and being coated, the skin is being coated by the microbes that are present in the vagina. The newborn is, with luck, will be breastfed, and breastfeeding itself promotes a lot of growth of the right kinds of microbes. So that this is a sort of story that we're just getting into in the lab. But as well as that, it provides some microbes themselves as well. And so there are microbes that actually seem to translocate into breast milk in order for the newborn to be colonized by these microbes from the mother in a very effective way. And then, of course, as an infant gets a little bit older and starts crawling around the environment, starts to interact with that environment, and we've all seen infants pick things up off the floor and put them in their mouths, and it's kind of horrifying and terrifying for parents. And it used to be thought that that was just a sort of a tactile response to the environment to sort of feel their way around. And now it's kind of thought that maybe there's an evolutionary advantage to putting things in your mouth to colonize yourself with microbes that are present in your environment. And so through this time, from being a fetus to being an infant, and then through into life, there's a progressive microbial colonization, and that sort of finishes around about the age of 3 years old. Now, if we think about some of the things that we do in the environment that can play into this, we have a lot of lifestyle things that we do that can actually interfere with this process of microbiome acquisition. Now, I'm not suggesting that any of these are unnecessary. Some of them are life-saving. What I'm saying is that it's only been recently realized that some of these may actually interrupt or interfere somehow with microbial acquisition. And perhaps we ought to sit up and take notice of this a little bit more. And as I mentioned, the window for proper gut microbiota development is narrow. So by the age of 3 years, an infant has gained its adult-like microbiome. And then for reasons that are just becoming clear now to do with the immunology of the situation, that microbiome becomes set and quite difficult to change. And so if you end up with a poor microbiome or a low diversity microbiome, there seem to be channels in place that prevents you from sort of increasing that microbial composition diversity after the age of around 3 years. And that's something as well that's obviously translationally very important and a very interesting area of research right now. So there's a big idea in the literature right now, which seems to be becoming more and more true and more alarming, in fact, that we are actually eroding microbiome diversity. And as a result of that, what does that mean? So the missing microbiota hypothesis came about through Marty Blaser and Stan Falco, who wrote a paper in Nature Reviews Microbiology in 2009, which was really the first kind of heralding of this era of wondering what we have done to our microbiome. Before that, there was the hygiene hypothesis and other hypotheses that kind of said the same thing, but this was the first time that the hypothesis was stated such that a loss of microbiota generally compounds over generations, and that recent lifestyle changes have greatly exacerbated this loss. And so if you look at this graph on the left-hand side, what they're saying is that over a period of generations, and these are measured at 20-year intervals, microbiome starts off quite high, let's say, but through these lifestyle interventions, we're losing microbes at a rate that is unable to be compensated for by potential horizontal acquisition, in other words, from the environment. And so that is a big problem, and something that was really just put out there as a theory. But since then, there have been many papers that have come out that have actually shown that it seems to be very true, and, as you imagine, quite alarming. And one of those is that there are many examples of diseases where we can tell that there are reduced gut microbiota diversity in published research. For example, there's inflammatory bowel diseases, colorectal cancer, and all of these other things that are on the screen. All of these are examples of diseases that are associated with low gut microbial diversity. Now, association isn't the same as cause. However, over the years, several of these diseases have been shown to be causal. So, for example, Clostridium or Clostridioides difficile infection is caused by a lack of diversity in the gut that allows the overgrowth of Clostridioides difficile, whereupon it starts to produce toxin and cause disease. And even colorectal cancer now, we're recognizing, is initiated by an aberrant gut microbiome that seems to be allowed to happen through low gut microbial diversity, and that's sort of very new research. And so the more that we look, the more that points to this idea of the missing microbe hypothesis. Another interesting point is that there's been studies that has been done, and particularly this one in Cell, that talks about migration from non-Western nation to the United States. And so this is a migration from Southeast Asia to the United States, and looking at the gut microbiome diversity of individuals over different generations. And so pre-immigration, it's been shown that the gut microbial diversity is very high, and particularly high in Prevotella strains, which have a lot of fiber-degrading enzymes, reflecting the much higher degree of fiber in the diet of these individuals. And then in new arrivals, we see immediately a drop in gut microbiome diversity to a certain extent. And in long-term residents, even more so. We see the same in second generation US-born individuals, and also European settlers from several generations ago. And this seems to also correlate with the degree of obesity in the population. And so this seems to be a very interesting kind of smoking gun, looking at this gut microbial diversity, but also it sort of serves to illustrate this point that diversity seems to be the underpinning of all of gut health. So another interesting report that came out recently that is related to microbial diversity is that we're starting to, this idea that health and disease markers correlate with gut microbiome composition. And this is a study that has been done on thousands of people. So we're really starting to see patterns with thousands of people. And I apologize, it's very difficult to read the writing. But what I want you to look at and focus on is the pattern. So on the bottom of this graph is actually a measure of diversity that we call Shannon diversity. And in the middle of the graph, that's where we see a change from negative to positive. And so what you can't read here very well probably on the left-hand side, but what I can tell you is that a positive microbial diversity, this positive Shannon diversity, is generally associated with beneficial things that you do. So for example, the number of times you eat fruits per week, or the amount of times you eat cruciferous vegetables per week, or oil, and a lot of things actually to do with diet. Whereas on the negative side of things, these are all related to counts of triglycerides and other scores like that, weight, and increased weight and lower diversity, as well as a number of other dietary things in a negative way. And so we're really starting to hammer this home now, hopefully, that we realize that more diversity in your gut microbiome seems to correlate with better health. So then that leaves the question, well, if my gut microbiome is less diverse than it should be, can I fix it? And so this is where we come into this idea of using probiotics and beyond. So can we fix these poorly diverse gut microbiomes using probiotics? So the idea might be, and you might have heard this even from a physician, if you take an antibiotic, then you can just cancel the negative effects out by using a probiotic. And you think that that sounds, reasonable, but actually that's not true at all. There are many, many different strains of probiotics, but generally there's only a very few species available. And available commercially. There are many manufacturers, some are legitimate, most are not, and that is partly because of the way that these things are moderated and regulated. And there are many, many overinflated claims as a result of that. These are food products, and as food products, they do not undergo the same types of rigorous claim testing, if you like, that a drug product would do. And there's actually very little actual science. It's very depressing when the more you look into this, how little science there actually is that is done on for example, the mechanisms of how a given probiotic might work and how what strains might be used for. And a study of, it's a fairly old study now, but still very true to this day, of 14 commercial probiotic products showed that many of them contained unadvertised additional lactobacilli and bifidobacteria. Others were missing species listed on the product label. Many probiotic capsules actually contain far fewer than the number of microbial cells advertised. And so when you see that they contain X number of billion cells in a capsule, that is at the time of manufacture. And then after that, that number dwindles very, very quickly. So the layperson's view of probiotics, I love this comic. It's this idea that you just go into the supermarket and you see all this live yogurt and kefir and stuff on the shelf, and everything's calling out to colonize your intestines, and isn't that great? But the problem is that that's based on a couple of myths. One of them is that probiotics found in food are the same kinds of species that are found in the gut, and that's just simply not true. They are similar species, but actually they're very, very different to each other in the way that two strains of E. coli can be very different to each other. One can be a probiotic, one can be a pathogen. So just because they share the same species name doesn't necessarily make them probiotics. The other myth is that probiotics colonize the gut. In fact, they don't, and many of them find it very, very difficult to survive in the gut environment and die very fast upon taking them. So my view as a microbiologist, my view of probiotics is a little more like this, that we have the normal gut microbiota. There's around 100 billion to 1 trillion colony forming units per mil, versus little Lego man probiotic with 2 to 15 billion CFU per capsule, but hardly any of that will make that through down into the gut. And so really they don't stand too much of a chance. And now I'm not saying that all probiotics are bad. There are some which have some proven clinical benefits. And I think that most of those proven clinical benefits are not through colonization, but again, through the production of certain molecules that might be more small molecules that might have health beneficial effects. And so this is a sort of an area of current research. But ecobiotics are kind of the next generation probiotics. There's actually a lot more to the gut microbiome than the very few species that are currently used as probiotics. And those of you that do use probiotics probably know that, most of them are made up of bifidobacterium, lactobacillus, and not much else. Ecobiotics, on the other hand, is this sort of new realm where we're looking at whole ecosystems derived from microbiomes of supremely healthy individuals encapsulated into capsules like this. So rather than just one or two species, now we have tens or even perhaps even hundreds of strains that we can deliver to an individual as a whole ecosystem. And the benefit of that is that some of those beneficial strains within that ecosystem will be helped along by the other strains within the ecosystem that act in a supporting role. The trouble is, we don't really know what to replace when you've got a so bad microbiome. What does that mean? And that's a very, very difficult question to answer. So do we know whether microbiomes derived from healthy people, for example, from industrialized nations, are as healthy as they could possibly be? And what about these missing microbes I've told you about? So which microbes will make you healthier? So I've got into this field recently because I'm very interested in this idea of vanish microbes, which I'll get to in a second. So how do we know that people in the industrialized world have a reduced gut diversity? Well, we can't go back in time to look at these microbiomes before the time of antibiotics and refined foods and all the things that we've introduced into our lifestyles. But what we can do is we can look at indigenous peoples who haven't had exposure to those things. And there are very small pockets of these individuals that still live today around the globe. And from DNA profiling, their gut microbiomes are far more diverse than ours are. And so we really think that we're onto something here. These are rich in vanish microbes, and vanish stands for volatile or associated negatively with industrialized societies of humans. On the other hand, we have blossom microbes, which are found much more often in industrialized populations, and that stands for bloom or selected in societies of urbanization or modernization. So you might see that, that term used as well. So why don't we just then culture microbes from these indigenous peoples? Well, that way we can better understand what we're missing. But the problem is that that's actually quite difficult to do. Now, one of the reasons is that these populations of peoples who have been sort of cut off from the industrial world, quite, as you might imagine, are in very remote areas and sometimes very difficult to access. There's dangerous terrain, there's endemic disease, there's culture and language barriers, there's often a lot of political turmoil in the countries in which they reside. And that means that there are also ethical challenges as well. And it's also just on a practical, from a practical point of view, very difficult to go to these remote areas and preserve samples so that we can actually start looking at the gut microbiomes within them. If you want to keep the gut microbes alive, you have to keep them cool and free of oxygen, and that can be very tricky to do. However, we have a bit of a secret weapon in my lab. Where do we start? We start with David Good. He's a man with a unique family heritage. His story starts here. This is a book written by his father, an American anthropologist who went into the jungle in the Venezuelan jungle, the Amazon, in the 1970s for 6 months to do a PhD project and stay for 12 years. And he ended up marrying a Yanomami woman. It's a very long story I don't have time to go into here. The product of that relationship is David. And David actually was born and raised in the United States. He, a number of years ago, found his mother, who'd returned to the jungle not long after his birth, and actually managed to find him in the jungle. And so this is also a fabulous story. The reason that I'm bringing David up is because his story is continuing here at Guelph. He's now my PhD student, and he's a biologist as well. And so he is a very interesting man and very interested in microbiomes. He approached me about doing a PhD, and I heartily agreed because his research aligns very well with what I'm trying to do with vanish microbes. So David is a Venezuelan citizen as well as an American citizen, which makes him quite unique for a number of reasons, but also because he is a half Yanomami man, he has the ability to actually interact with the indigenous peoples in Amazona or Amazonas. And he is unique within that ability, because it's very difficult to actually gain access to these people and do it in an ethical way. So David, a number of years ago, started something called the Good Project. And the Good Project is a non-profit dedicated to help support the future of the Yanomami people, really by doing a lot of research on the one hand, but also making sure that the profits and the proceeds of that research and that understanding go back to directly benefit the Yanomami people, who are in fact, of course, his family. There's a map there. You can see that the Yanomami territory in green straddles the border of Brazil and Venezuela, and David's family will actually live on the Venezuelan side of the border. So David, back in 2019, went on an expedition where he actually was able to take some poop samples onto banana leaves from his family, put them into Eppendorfs and put them in these little Nero packs that we had. He was able to put those in a cool box that was solar powered. There is no power. He's really very much off the grid when he's out with his family in the jungle. And then he was able to trek that back then on a series of boats down the Orinoco River to Esmeralda, the nearest town, where he could take a small plane to a larger plane. And then the samples eventually made it to the University of Guelph. It was a very long and very scary journey for me, and also for David, because every time he goes to the jungle, he is risking his life. Although he's going to see his family, it's a very dangerous expedition that he does. He doesn't do it alone. He has a whole group of expeditioners that he goes with. And when he goes down to the jungle, it's not just to collect microbiome samples, but he's actually also doing some important work, delivering some much needed medical supplies and helping with Yanomami education and trying to give them a seat at the table in terms of the political situation in which they find themselves. So that has allowed us to overcome some of the barriers, the remoteness and difficult access, the culture and language barriers, of course. David speaks Yanomami and also is fluent in English, of course, and Spanish, as a matter of fact. Gets over the ethical challenges because David is doing this in a supremely ethical way. And it also helps us with preserving the samples. We've shown that we can do it this way. I'll show you in a minute that we've actually been able to culture a lot of microbes from this. What it doesn't help with is the political turmoil. Venezuela is a failed state. It's extremely difficult to get hold of things like gas and permits and all those kinds of things. Very, very difficult to do, but we are doing it. And of course, it's very dangerous terrain and there's a lot of endemic disease. And David actually came back this last round with a nasty bout of malaria, which he had to treat in the United States. Just to show you some family pictures, if you like, of David. This is when he's in the jungle. David with his family. And this is also an image from Wikimedia that shows you sort of an aerial view of one of the houses in which these people live. So they don't live in individual houses, they live in these chaponos, which are large dwellings with a little bit of shelter and a clearing in the middle. And the whole family, the whole extended family lives within these under this sort of shelter, and each has their own hearth, their own little fire that they have, and that's what creates the family unit. All right, so now the sample safely arrived. Well, then now what? So it turns out that in my lab, what we're very good at is growing and culturing gut microbes. And so the human colon is a sophisticated bioreactor. And so the way that we do this is we grow these microbial ecosystems as a whole in chemostat bioreactors that we use to approximate the human colonic environment. And we do that from adjusting a number of things that we can to kind of make it very similar to the environment in the colon. This is what it looks like. We have many of these systems now. This is a 6-vessel system with like 4 vessels up and running. And my lab is actually renowned for its ability to culture very fastidious gut microbes. To do this, we use this thing, we call it the robo-gut, actually, we've dubbed it the robo-gut. And we found that if you seed this system with fresh feces, then it supports broad ecosystem growth for several weeks. And that includes bacteria, fungi, archaea, viruses, all of the things. It's a host-free system. It's not a perfect model, but we can add some host components to see what those might do to the ecosystem, for example, things like secretory IgA. And we can test the effects of stresses on the ecosystem. And because it's in vitro, no one gets hurt. So it's really great. We don't have to get ethical consent to do anything once we have the microbes in place. And more importantly, we can easily measure the metabolic output by just withdrawing a sample from these glass and stainless steel vessels whenever we want to and measuring what those metabolites might be. And that's important because, as I mentioned, it's not really about what species are there. it's about what the species are doing that's important. So what we did is we made a bioreactor feed, the media designed to mimic the Yanomami diet. So it's actually quite a bit different from what we do for the industrial or for Western diets. And that we hoped would allow us to recover a large number of species. And so the biggest difference there is it's very high fiber and from very specific foods that we don't generally eat very much in the Western world. For example, cassava, plantain, hearts and palm. Also a lot of fish protein and chitin. They eat a lot of mushrooms. And what we hoped is that, that this diet would support more Yanomami microbes than a Western style diet. Sarah in my lab as well, it's an absolutely amazing student who I then recruited as my lab technician, also performed isolations on a lot of very, very specialist media to try to isolate some of these microbes, many of them for the very first time. And so here is a graph just to show you what should we find. So on this graph on the right-hand side, the leftmost bar is actually a sample from a Westerner. So this is the type of metagenomic analysis we would expect from a fecal sample from a Westerner. You can see it's not particularly diverse. This is at the phylum level, so a very high level of taxonomy. And what you see on the rest of this right-hand side is the 9 samples we brought back. You can see that they're very much more diverse than the gut microbiome of this healthy Westerner. So this was all done with something called shotgun metagenomics. Instead of just looking at 16S here, we're actually looking at everything. And so we're also looking at the, although I'm just showing here the bacterial stuff, we're also looking at the fungi and the protists and the viruses and everything that's in there. And it's, of course, as I mentioned, the Western microbiome is different from the Yanomami microbiome. And DNA profiling gave us a really good snapshot of the species present that we could target. So, of course, I've just shown you here at a very high level, but we can go down more deeply than that and look at the species that we might expect to see there. But what it doesn't do is a metagenomic sequencing doesn't distinguish dead microbes from live ones. So far, what have we been able to grow? So again, a horrible looking figure, I'm afraid. It's just very complicated, but let me just talk you through it. This is just a phylogenetic tree that shows the sorts of species that we would expect to find from one of the samples. And what Sarah has done has labeled them with little dots. So there's a blue dot if they have been sequenced. So in other words, if we detected them in the sequencing survey that we did with metagenomics. And there's a red dot next to the name if we actually cultured it as well. So she's done a pretty good job. And in fact, this is a lot more than we expected because, again, we didn't know whether or not these microbes would survive their journey out of the jungle. What we found was that we've isolated many microbes. So far, we have we've just done 4 Yanomami samples of the 9 that we retrieved. It's a very long-winded, very high stakes job, so we'd like to do it properly. And we have at least 317 different species, including microbes representing lots of novel species. And if you contrast that to the previous study that was done on the Yanomami people, they only found 27 unique species from 12 fecal samples using 7 media types. So we've really done a much deeper dive. And that's just to kind of give you a slightly bigger for those that can't see too much on a small screen. So one of the things that we found was a novel species of treponema from the human gut. So far, treponema have only been seen in hunter-gatherer people from around the globe and only by looking at DNA samples. They've never been seen in Westerners, and that's what the image at the bottom is showing. There's a big black box for the USA. We don't see these types of treponemes in individuals from the USA, but we have been able to now isolate it. So this is representative of a missing microbe. We think this might be one of the poster children for a missing microbe, in fact, and so we're doing a lot of studies on this. We asked the question, can we improve industrialized world gut microbiomes with a Yanomami diet? So for example, if we took a healthy Westerner's microbiome and we fed it a Yanomami diet, would we start to see more diversity? And that's partly because the Yanomami diet has a lot more fiber than a Western diet. We're asking the question, are these microbes present, but they're just so present at such undetectable levels that, that we think that they're not there. Well, when we did that, we just a quick snapshot of what we saw, but actually it's easier to look from this Venn diagram here. At the end of our run in the RoboGut on the Yanomami diet, we see a lot more diversity in this healthy Westerners microbiome compared to in the industrial world diet. So certainly diet does seem to support and allow microbes to bloom, which may just be kind of hanging in there in the gut of a healthy individual. But it's not nearly as diverse as a Yanomami fecal sample. So have we found our missing microbes? So in collaboration with a colleague of mine at Harvard, Alex Kostick, who looked at authenticated Paleo-feces, fossilized feces from individuals from around 1,000 to 2,000 years ago, he sequenced their metagenomes as well. And again, a horrible looking diagram. Let me walk you through it. On this left-hand side here, on the right-hand side of the graph are sort of separated by a white division, are all of the samples from the industrial world. And on the left-hand side are all the samples from non-industrial civilizations in Fiji, Tanzania, Peru, and Madagascar. And on the far left-hand side, these are the Paleo-feces samples. So you can see, hopefully, and this is just a presence or absence of graph of these species, that the species that are present in the industrial world seem to be quite different from those that are present in the non-industrial world. And the Paleo-feces matches the non-industrial world much better. And then just to point those out. And so what we did then is we decided to incorporate the Yanomami microbiomes in there. And instead of looking at presence absence of species, this time we looked at K-zyme meta-analysis. So these are carbohydrate active enzymes that are important for processing of dietary carbohydrates. And again, when we do that, we see on the right-hand side of this graph, rather, the industrial microbiomes. And on the left-hand side, we see the non-industrial microbiomes. And then on the very far left, we've got the Paleo-feces again and the Yanomami samples. And if I just show you those yellow ones there and blow it up a bit, hopefully what you can see is that the Paleo-feces and the Yanomami samples actually look incredibly similar to each other. And so the Yanomami ecosystems may represent a microbiome time capsule. And what we're hoping is that by culturing and studying these enigmatic ecosystems, we might actually understand what functionality we've lost and what we've gained through industrialization. Now, as I mentioned, the ethical considerations are very important. This work's being done under close collaboration with the Good Project. All necessary permits for obtaining samples are in place. David's obtained assent from his family to take part in the research, and expeditions to study the Yanomami microbiome are done in tandem with delivery of life-saving medical supplies. And all the PIs have signed agreements with the Good Project to ensure any and all intellectual property obtained from study of the microbiome belongs to the Good Project, and any proceeds from the sale of this IP will be used to support and benefit the Yanomami people. If you want to find out more about this project and the good work that they're doing, this is the website to go to. So in summary, we've achieved what we thought was once impossible, the isolation and culture of novel fastidious gut microbial species from a remote hunter-gatherer population. And as expected, there's a lot more diversity in these microbiomes than we see in the industrialized world. The missing microbes in the industrialized population seem to really be missing, not just present at undetectable levels. And that upholds the missing microbiome hypothesis. Some beneficial microbes increase in number when consuming a Yanomami diet compared to an industrial world diet. So what's next? What do these novel species found in Yanomami fecal samples contribute to the gut microbial ecosystem? So we'll carry on with our RoboGut experiments and then start adding novel species to microbiomes derived from individuals from the industrial world. We'll carefully characterize, as we're doing now, novel species, the genomes and their physiology. And we'll sequence the samples in situ. And this is something that David is planning to do next summer. He hopes to become the first person to sequence fecal microbiomes directly in the Amazon jungle off-grid using the Oxford nanopore system and a very sophisticated setup that he's got going in what we call the jungle lab in my lab right now, that he can actually operate off-grid using solar power. So hopefully we'll be able to show you more of that in the future. And this is just to acknowledge all the people in my lab. You can see David and Sarah at the front of this row. This picture was taken last week, so it's a very new one. And thanks to all the people that fund it and my colleagues at the Joslin Diabetes Centre. And with that, I will stop sharing. And I guess we can go to the question time.
Thank you, Emma. That was a fantastic presentation. I learned so much, and it was just so interesting to learn about the differences in the microbiome. There's so much that I never thought of and considered before. That was a really excellent presentation. So now we're going to move to the Q&A pod to address some of the questions that are there. Please continue to enter your questions into the pod if you have not already had the opportunity to do so. So I'm going to get started. There's a few already that are coming in. So the first question that we have here is, given the individual variability in our microbiome, do you feel that probiotic supplementation and therapy will go in the direction of precision medicine? Or do you think there is still possibility for probiotics that are beneficial for almost everyone?
Yeah, that's a great question. I actually don't think there is such thing as a probiotic that's beneficial for everyone, because everyone has a different gut microbiome. And I really do think that precision medicine is the way ahead in terms of making, bespoke microbiomes or microbial ecosystem therapeutics that actually are designed to help a given individual. We're not there yet. It would be an incredibly expensive thing to do, for one thing. But I think the more that we look, the more that we're beginning to find groups of microbes, or guilds, if you like, that have beneficial properties that we can put together in groups and allow them to sort of and then put them together to make a bespoke medicine that we might be able to use to help people in the future. That's some way off yet.
Thank you. So the next question that we have is, would you recommend pre or probiotic enhanced infant formula for non-breastfed infants?
Oh, that's a really great question. So I'm not sure is the answer. I think I probably wouldn't recommend probiotic enhanced, just because I'm not sure that the right probiotics are in place right now. What I have seen as well is in terms of prebiotics, formula milk is being supplemented with what we call human milk oligosaccharides, which are the sugars, the types of sugars that we actually find in breast milk. But unfortunately, we're certainly not doing enough there, because there are over 200 different types of human milk oligosaccharides, and these companies, and they're very complicated sugars, very difficult to synthesize, very expensive. And so the companies have limited themselves to the ones which are cheap to produce and easy to produce. And unfortunately, those are not the ones that tend to support the gut microbiome. And we have some new data that's not published yet that shows that. So I think that formula milk has a long way to come. I think that it's certainly better than no milk at all, and breastfeeding is a great thing to do, but not everyone can do it. So I do think that it's certainly better than it was, and it's improving. I think we can do a lot better. So I think that these companies need to step up and try to do that.
Thank you. Now we're going to move into a couple of questions related to antibiotics and the microbiome. I'm going to try to tie some of these in together. So to what extent do you think antimicrobials, for example, penicillin, for treponema, have impacted industrialized microbiomes? Or is it more the other effects of the industrialized society that are causing more of the impact here?
Great question. We don't know. We can't go back in time really to see that. But what we can do is start to now take these gut microbiomes that contain treponemes from these Yanomami individuals that we have, and we can start in the robogut to expose them to the types of exposures that we would have had in the industrial world. So for example, penicillin, that's a very good point. That's how treponema infections are generally treated. Not necessarily, not all treponemes are bad, right? So what would penicillin have done? So we do think that it's a mixture of many things, and it's very difficult to say which of those things has had the biggest effect. Certainly the two contenders that have had the most effect would be a change in xenobiotics, so antibiotic use, so availability of those things, and also dietary changes for sure. So trying to understand the importance of those two things is one of the things on our list of things to do, actually.
And then, related to the poll question you had for clindamycin, there's some questions that are coming up of how can you use the information, how to counsel patients on taking clindamycin, and what is the impact of antibiotics such as clindamycin for the degree of impact they have on the microbiome as well? Is it like 10%, 90%?
Yeah, very difficult and difficult to measure because everyone has a different microbiome. So everyone's microbiome is going to be differentially sensitive to something like clindamycin. What we do know is that clindamycin is one of the worst offenders in terms of clearing out gut microbes. But that's because it's such a good antibiotic and it's very penetrative, and it's often used in dental infections and other infections which are otherwise difficult to get at. It's a life-saving drug, and I would never sit here and say we should stop using antibiotics. I think that would be crazy. What we need to understand is can we provide these antibiotics in slightly better ways that help to preserve the integrity of the gut microbiome. So oftentimes clindamycin is given, but it's not given for a gut infection, it's given for something else, usually a dental infection, as I said. So are there ways that we can actually prevent this drug from finding its way to the gut microbiome, or to limit its ability to do so? I can't remember the beginning of the question, but was it something about, are there other antibiotics that we can use, or?
The question is just more so related to how, kind of the impact that and clindamycin being as an example, the impact of antibiotics on the microbiome.
Yeah, so I think, I picked on clindamycin because clindamycin is by far associated with more incidences of antibiotic-associated diarrhea and potentially Clostridioides difficile infection than any other antibiotics, as far as I know. I mean, I'm not a clinician, there might be some others, but that's by far the one that's most commonly administered. And so there are antibiotics, of course, all different antibiotics have different mechanisms of action, different pharmacology, different ways in which they're sort of spread around the body. So those that, as I mentioned, the more that we understand this, the more that we're realizing that these microbes need to be protected in the gut from the ravages of antibiotics. I think we can have our cake and eat it too.
I like that. I hope we can get some diverse microbiome from cake eating as well. So there's some questions related to diet. So, and I'm going to again try to put some of these together. So, kind of what would you recommend for promoting a microbiome diverse diet, and is there something that would be more obtainable for those living with limited incomes in Ontario?
Yeah, that's a very, very good question, and I'm not entirely sure that I can answer that appropriately, but I'll do my best. So I think that one of the best things that we can do is to make people realize that the best way to get the nutrients that they eat is through eating fresh food that isn't processed. And I think that's becoming clearer and clearer. Of course, it's very difficult to tell people of low incomes to just eat fresh food because that can be much more expensive than processed food. And so that is something that we need to address as a community. And I'm not, I wish I knew the answers to that. I think that one of the key pieces of information that we need to get across to people is that we need to eat a lot more fiber than we actually do, and we also need to stop eating anything which has additives in them. And what I mean by additives, I mean artificial additives like artificial sweeteners, colors, preservatives, all of those kinds of things, because now we are starting to realize that those types of compounds, although they've shown to be sort of safe for human consumption, have never been tested until recently for their effects on the microbiome. And that actually, their effects on the microbiome can sometimes be fairly far-reaching. And in some cases, for example, some of the artificial sweeteners can actually cause a type 2 diabetes type microbiome, which is the very disease that they're sort of introduced to try to fight. And so until we can understand what these types of foods are going, can do, we've got to be very careful about how we administer them. So I think the answer is a tricky one. As I said, it's a socioeconomic one. We need to encourage people to eat more fresh food and to make food from scratch, but we need to make that we need to make that approachable and attainable for people on low incomes, because that is, it's not, it's often with that, outside of people's grasp. So yeah, I wish I knew the answer.
That is a good answer. Thank you. So we have 3 minutes left. I'm going to ask very quickly one last question. Is there any data related to intermittent fasting and what that impact has on your microbiome?
Yeah, and in fact, we can actually model that in the lab, and we have been doing so. So the microbiome is obviously constantly fed by what we're eating. So what happens when we stop eating or we fast intermittently? Well, it turns out that mucin, which we produce endogenously, regardless of whether we're fasting or not, is a very major food source for the gut, for the microbes that live in our gut. It's the kind of primary food source. When you fast, you are not killing your microbes, far from it. You're actually, in some cases, you may just be allowing some of the microbes that are very fastidious and have very particular dietary needs to bloom a little bit, because it allows them to maybe some of these are mucin degraders. So we're starting to understand that that there is even a diurnal kind of pattern to the types of metabolites that are being produced by the microbiome, even in people who don't fast. And so wondering, what's happening in people that fast is a really interesting question. I don't think anyone knows the answer yet, but we do know that it does something. And that it seems to do something beneficial. It's just very difficult to then kind of sort of propose that we do this blanket across the whole population, because everyone is different. And so understanding the metabolism and everyone's unique microbiome, I think, is the first step. And then we can kind of see, well, what might happen in a given individual if we started to introduce intermittent fasting.
Thank you for that response. We have so many more really great questions, and I feel like we could sit here and ask you the questions all day long. I mean, I have certainly a very long list of questions that I didn't get to touch upon. So thank you, everyone, for all of your excellent questions. So as we wrap up today's PHO microbiology round session, I would like to thank Emma Allen-Vercoe for presenting. I'd also like to thank everyone who joined us for today's session. We had a really great turnout, so thank you. You can expect to receive a brief and anonymous PHO microbiology round survey for today's session. Please try to complete this to help us improve our programming. Lastly, to access past PHO rounds presentations and view confirmed upcoming sessions, please visit the PHO website. Head to Education and Events and click on Presentations. Thank you, everyone, and have a wonderful day.